I sometimes wonder why anybody talks about anything else, because this is the most interesting topic there is. Where do we come from? How did we get here? What brought us into existence? What is our relationship to reality as a whole?
You look at the incredible diversity and complexity of life, and inevitably the question arises, what brought all this into existence? Was it simply chance and necessity, undirected natural forces? Or is there something else going on? Is there a purpose, a plan, a design due to an intelligent cause? I think that is the fundamental question.
The scientists who came to Pajaro Dunes set out to re-examine the mystery of life's origin. For each had significant doubts about widely held evolutionary ideas. Among them, biochemist Michael Behe questioned how natural processes could have assembled the intricate structures found within living cells. Dean Kenyon was an evolutionary biologist who no longer thought that chemistry alone could account for the origin of life on Earth. And Stephen Meyer, Paul Nelson, and William Dembski were seeking a new approach, one that could explain the origin of the genetic information encoded in living organisms. These scientists and philosophers began to formulate an alternative to the central theory of modern biology, a theory born in the mind of a British naturalist. His name was Charles Darwin.
It's really interesting to notice that the more we know about life and the more we know about biology, the more problems Darwinism has and the more design becomes apparent.
Since 1988, Dr. Michael Behe has investigated complex biological systems that seem to defy explanation by natural selection. Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
There are as many molecular machines in the human body as there are functions that the body has to do. So if you think about hearing, seeing, smelling, tasting, feeling, blood clotting, respiratory action, the immune response, all of those require a host of machines.
When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
But could Darwin's small, favorable variations have produced a bacterial flagellum? Some scientists doubt the possibility.
How could something new, like a bacterioflagellar motor and all the components that go with it, how could it develop out of a population of bacteria that don't have that system?
If you had a bacterium with a tail that didn't function as a flagellum, chances are natural selection would eliminate it. The only way you can select for a flagellum is if you have a flagellum that works, and that means you have to have all the pieces of the motor in place to begin with. So natural selection can't get you the bacterial flagellum. It can only work after the flagellum is there and operating.
Bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually you're going to have to account for the function of every single part is originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity, for its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction of a house, where workers follow a detailed blueprint and plan for assembly. The foundation of a house is poured before the walls are erected. Plumbing and electrical fixtures are installed prior to enclosing the walls of the structure. Windows must be hung before siding is applied, and shingles are attached only after plywood sheets are nailed to the rafters. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now. A rod is added, a ring is added, another rod is added, a U-joint's added. Once the U-joint's at a certain size and a certain degree of bend, about a quarter turn, that's shut off, and then you start adding components for the propeller. These are all made in a precise sequence, just like you would build a building.
To build a motor correctly requires a complex system of machines that coordinate the timing of the assembly instructions. But how could natural selection construct such a system?
The co-option argument doesn't explain this. You see, in order to construct that flagellar mechanism, or tens of thousands of other such mechanisms in the cell, you require other machines to regulate the assembly of these structures. And those machines themselves require machines for their assembly.
If even one of these pieces is missing or put in the wrong place, your motor isn't going to work. So this apparatus to assemble the flagellar motor is itself irreducibly complex. In fact, what we have here is irreducible complexity all the way down.
We know a lot about the bacterial flagellum. We still have a lot to learn, but we know a lot about it. And there's no explanation for how this complex molecular machine was ever produced by a Darwinian mechanism.
By the 1960s, scientists had determined that even simple cells are made of thousands of different types of proteins, and the function of these molecules derives from their highly complex three-dimensional shapes. The irregular shapes of some proteins allow them to catalyze or trigger chemical reactions because of the hand-and-glove fit that they have with other molecules in the cell, while other protein molecules form interlocking structural components.
The individual parts of a bacterial motor, like this ring structure, are each made of either a single protein molecule or an assembly of proteins fitted together into a specific shape. These proteins are, in turn, made of smaller chemical units called amino acids that are linked together in long chains.
There is a very great degree of intricacy of architecture down in the cell units in these protein-forming amino acids.
In nature, 20 different types of amino acids are used to construct protein chains. Biologists have compared them to the 26 letters of the English alphabet.
Alphabetic letters can be arranged in a huge number of possible combinations, and it's the sequential arrangement of the letters that determines whether you have meaningful words and sentences. If the letters are arranged correctly, you'll get meaningful text. But if they're not arranged correctly, you'll get gibberish. And the same principle applies for amino acids and proteins.
There are at least 30,000 distinct types of proteins, each made of a different combination of the same 20 amino acids. They are arranged, like letters, to form chains, often hundreds of units long. If the amino acids are sequenced correctly, then the chain will fold into a functioning protein.
proteins are arranged with their amino acids in such a way that the amino acids collapse on each other into an architecture that is pre-programmed by the order of the amino acids. It folds into a certain structure, and that structure can do a certain function. So all proteins in the cell have a certain three-dimensional pattern that's based on the arrangement of amino acids in the chain.
This arrangement is critical. For if the amino acids are incorrectly sequenced, a useless chain forms, and instead of folding into a protein, it will be destroyed in the cell.
Proteins, like written languages or computer codes, have a high degree of specificity. The function of the whole depends upon the precise arrangement of the individual parts.
But what produces the precise sequencing of amino acids that gives rise to the specific shapes and functions of proteins? In living cells today, chains of amino acids are not formed directly by forces of attraction between their parts, the scenario Kenyon envisioned on the early Earth. Instead, another large molecule within the cell stores instructions for sequencing the amino acids in proteins. It is called DNA. To understand why, consider the difficulty of generating just 2 lines of Shakespeare's play Hamlet by dropping Scrabble letters onto a tabletop. Then consider that the specific genetic instructions required to build the proteins in even the simplest one-celled organism would fill hundreds of pages of printed text. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
When I look at molecular machines, or the incredibly complex process by which cells divide, I want to ask, is it possible that these things had an intelligence behind them, that there was a plan or a purpose to this structure? Science ought to be a search for the truth about the world. Now, we shouldn't prejudge what might be true. We shouldn't say, I don't like that explanation, so I'm going to put it to one side. Rather, when we come to a puzzle in nature, we ought to bring to that puzzle every possible cause that might explain it. One of the problems I have with evolutionary theory is it artificially rules out a kind of cause even before the evidence has a chance to speak. And the cause that's ruled out...
Consider, for example, these hieroglyphic messages carved upon the ruins of Egyptian monuments. No one would attribute the shapes and arrangements of these symbols to natural causes like sandstorms or erosion. Instead, we recognize them as the work of ancient scribes, intelligent human agents. Similar reasoning leads us to conclude that the mysterious stone figures on the shores of Easter Island were not formed by the actions of wind and water over great periods of time. Nor do we presume that plants could grow into these familiar shapes without some manner of intelligent guidance.
Of course, we make these inferences all the time, and we know they're correct. But the question is, on what basis do we make these inferences? What are the features that enable us to recognize intelligence?
On a seashore, another improbable pattern etched into the earth illustrates how we detect design. No one would infer that this message was written by the movement of the tides. Instead, because of the characteristics of this pattern, we identify the words as the products of intelligence.
That improbable arrangement also conforms to an independently given pattern, namely the shapes of the letters that we recognize from English alphabet and the words that we know from English vocabulary. And so it's the improbability of the arrangement plus the fact that it conforms to an independently given pattern that triggers the awareness of design.
To me, the great promise of design is it gives us a new tool and explanation that belongs in the toolkit of science. Intelligent causes are real, they leave evidence of their existence, and a healthy science is a science that seeks the truth and lets the evidence speak for itself.
The argument for intelligent design is based on observation of the facts. Now that's my definition of good science. It's observation of the facts. Now when you observe the facts, as Michael Behe has done, what do you observe? You observe this incredible pattern of interrelated complexity.
And the way we conclude intelligent design for the bacterial flagellum is the same way we conclude intelligent design for an outboard motor. When we see an outboard motor, we see the way the parts interact and so on. We know somebody made that. The reasoning is the same for biological machines. So the idea of intelligent design is a completely scientific one. Certainly it might have religious implications, but it does not depend on religious premises.
When I look at the evidence objectively, without ruling out the possibility of design, design just leaps up as the most likely explanation. And that's why I believe that it's true.
I think design is back on the table. You know, we can't explain these systems by natural law. And if we're searching for truth, and they are in fact designed, if we have to be design engineers to understand them, then I say, what's the problem? You know, you go where the data leads you. And the implications, yeah, they have profound metaphysical implications, but so be it.
So it's a powerful idea that the universe is rational and comprehensible, underwritten by a supreme intelligence that meant for this world to be understood, is something that underwrites then the program of science, because then you can go out and look at the world and the world will make sense. If it's all just a chaotic assemblage, there's no reason to expect any rationality out there. But if it in fact is the product of a mind, then you can go out and science becomes this enormous, wonderful puzzle-solving project in which you can expect to find rationality and beauty and comprehensibility right at the foundation of things.
150 years ago, Charles Darwin transformed science with his theory of natural selection. Today, that theory faces a formidable challenge. Intelligent design has sparked both discovery and intense debate over the origin of life on Earth. And for a growing number of scientists, it represents a paradigm, an idea with the power to, once again, redefine the foundations of scientific thought.
During the 19th century, scientists believed that there were two fundamental entities, matter and energy. But as we enter the 21st century, there's a third fundamental entity that science has had to recognize, and that is information. And so as we encounter the biology of the information age, the suspicion is growing that what we're seeing in the DNA molecule is actually an artifact of mind, an artifact of intelligence, something that can only be explained by intelligent design.